and all. Today we are moving for one more new lecture, that is PCR, and also in connection with in continuation with those things, we are also going to learn few types of molecular markers like Rflp, RAPD, and SSR markers, their importance, procedure, and application part. So coming to PCR, polymerase chain reaction. In the mid 1980s, Carey Mullis, a scientist, devised a method of replicating the genes, which is called as PCR. He was given the Nobel Prize for that time, and it was thought to be one of the revolutionary thing in the history of science. A DNA sequence less than one part in a million of the total sample can be cloned in this machine. So, in fact, a single gene can be amplified into a million copies of a duplicate. So just like a Xerox machine, we make 100 copies out of it. Similarly, this kind of machine can make copies of DNA or the sequence of genes. In order to determine the exact DNA sequence of a gene or the section of a DNA, it is necessary to have adequate sample of a particular gene to work with. So this is why PCR is of so valuable resource, because it allows a researcher to replicate a gene into a workable amount. Then coming to what are the different steps involved in this PCR. So PCR works on the basic principle of Peltier effect, and then powerful technique of amplifying the DNA involves cycling of the temperatures. There are three steps in this PCR. First step is denaturation, annealing, the second step, and the third step is elongation. The amplified DNA is separated by gel electrophorese. So the first step, that is denaturation. The DNA is denaturated at an elevated temperature, reducing it to single strands. Actually, DNA is a double-stranded thing. Because of this denaturation, it is going to separate into two single strands. Then annealing, this is the second step. Then cooling allows the primers to go and anneal to the complementary regions in this step. The third step is elongation. The DNA polymerase synthesizes a complementary strand in this elongation step. The result is a dramatic amplification of the DNA that exists between the primers. These cycles are repeated 20 to 40 times, each cycle providing two new templates for the next cycle. The amount of amplification is 2 raised to the power of n, that is n representing number of cycles that are performed. So this is a picture of a polymerase chain reaction thermocycler. This is called as a thermocycler. So after 20 cycles, this would give approximately 1 million fold amplification. After 40 cycles, the amplification would be somewhere around 1 into 10 to the power of 12. So the reaction is performed in the thermocycler, which is a programmable heating block that will cycle between melting, annealing, and polymerization temperatures n number of times, which is programmed as per. Then the polymerase reaction is very sensitive to the levels of divalent cations, especially magnesium ions and nucleotides, and the conditions for each particular application must be worked out. Then primer design is also extremely important for effective amplification, and the primers for the reaction must be very specific for the template to be amplified. So the cross reactivity with the non-target DNA sequences will result in non-specific amplification of DNA, which will be a useless thing. Also, the primers must not be capable of annealing to themselves or each other, as this may result in a very efficient will curb the results or will spoil the results of efficient amplification and resulting in the short nonsense DNAs. The reaction is limited in the size of DNAs to be amplified, and the most efficient amplification is in the 300 to 1000 base pair range. However, amplification of the products up to 4 kb has been reported up to now. Also, Taq polymerase has been reported to make frequent mismatch mistakes when incorporating new bases into this strand. So the most important consideration in PCR is contamination. If the sample that is being tested has even the smallest contamination with the DNA from the target, the reaction would amplify, that is multiply this DNA and report a falsely positive identification. So the PCR reaction mix, what does it contain? The target or the template DNA, thermostable DNA polymerase, one or more oligonucleotide primers, reaction buffer containing cofactor Mg+2, and DNTPs, dinucleotide phosphates. Then the reaction buffer then followed by these steps, three steps, that is denaturation, annealing, and elongation. Denaturation, it occurs for a time span of 2 to 4 minutes at 94 to 96 degrees centigrade with the strands separate. Then annealing 30 seconds, 44 to up to 4 minutes at 50 to 65 degrees centigrade this happens, and then followed by elongation 1 to 4 minutes at 72 degrees centigrade. So first of all, denaturation, separation of the strands into two single strands, then annealing, primers go and adhere to the complementary parts of the strands, then followed by extension, this next step where complementary DNA synthesis happens. So this is how it exponentially continues. At the end of 30 cycles, it's going to multiply to a million, and then 35, 36 cycles, somewhere around 68 billion copies. Then, so at a stretch, amplifies a subset of a genomic fragments of a particular gene, template DNA, which is heated, denaturation, two strands separate, 94 degrees centigrade, annealing of the primers, 50 to 60 degrees centigrade, then extension of the primer, 70 degrees centigrade, and this is repeated again and again number of times, and then the products are subjected to electrophoresis. And this is how it works, multiplication works. Then coming to our new topic, molecular marker. It's a DNA segment that is readily detected and whose inheritance can be easily be monitored. The use of molecular markers is based on the naturally occurring DNA polymorphism. A DNA marker is a small region of the DNA showing sequence polymorphism in different individuals within a species or among different species. The DNA markers are most widely used type of markers, predominantly due to their abundance. We have a number of types of markers, which may be biochemical markers, morphological markers, but however, DNA markers are of abundant use. The first such DNA markers to be used for the fragments produced by digestion with the restriction enzymes, restriction in the nucleus, which are also called as molecular scissors. That means the restriction fragment length polymorphism based genetic markers. So a wide range of molecular techniques are now available to detect polymorphism at DNA level. These are grouped into two based on the PCR technique. Non-PCR based approaches, example Rflp, PCR based approaches, example RAPD, AFLP, SSR, etc. So what are the ideal characteristics of a genetic marker? They should be highly polymorphic, codominant in the expression. What is codominant in the expression? That is nothing but it should differentiate between a dominant homozygote and heterozygote. Then frequent occurrence in the genome. Then selective neutral behavior, easy and fast assay. Then high reproducibility, it should replicate number of times, the result should not change. Easy transferability of the data across the labs. So we are doing something at a certain lab, we are getting some kind of result. That is when it is performed at a different lab, it should replicate, but it should not vary. Then genome specific in nature. Then economic to use, should be cost effective and amenable for multiplexing. What is this multiplexing? I will let you know shortly. And ability to be automated in the sense which can be subjected to the machine, so that machine does everything. So these are the few steps, few characteristics of a genetic marker which should be ideal for a genetic marker. Then all these steps, whatever we have discussed, will not be true for a single marker. Each one have their advantages and disadvantages. The first one is a restriction fragment length polymorphism. So this technique was first developed in 1980 and it is based on southern hybridization technique. So you know very well what is southern hybridization technique. So we'll let you know very shortly again. To refresh you again, then Rflp is used to describe the variation in the length of the fragments obtained from digestion of the DNA from two or more organisms with the same endonuclease. Then the variation at the DNA level is assayed by shearing the entire DNA with the restriction enzyme. Shearing in this is cutting. Then the restriction sites for a particular enzymes are present at several places throughout the entire genome, with the result that a large number of fragments of the DNA are produced. So the length of each segment depends upon the distance between two adjacent restriction sites. The plants are able to replicate the DNA with high accuracy and rapidity, but many mechanisms causing changes in the DNA are operative. Simple base pairs changes or large-scale changes as a result of inversions, translocations, deletions, or transposons may occur. So this will result in loss or gain of recognition site, and that in turn leads to restriction fragments of different lengths. And that becomes a starting point for our polymorphism or identification. These restriction fragments of different lengths between the genotypes can be detected on these southern blots and by using a suitable probe. So an Rflp is detected as a differential movement of the bands on the gel lanes from different species and strains. So each such band is regarded as a single Rflp locus. The pattern of Rflp generated will depend mainly on the differentiation of the DNA in the selected strains, restriction enzymes what we have used, and the DNA probe employed for southern hybridization. So what are the basic steps in Rflp analysis? First, isolate the DNA, then cutting the DNA into smaller fragments using restriction endonuclease enzymes, then separation of the fragments by gel electrophorese, transferring of these DNA fragments on to a nylon or a nitrocellulose membrane filter, and then visualization of the special DNA fragments by using labeled probes, and finally analysis of the results. So these are the steps. So number one, extracting the DNA, then cutting with restriction endonucleases, then subjecting those to gel electrophoresis, separated DNA fragments on gel agarose gel, then they are subjected to southern blotting. DNA is transferred onto the membrane. Further, these are attached to the radioactive probes, then remove the excess probe, then wash, then what you call membrane DNA with the covalently bonded probes is exposed to X-ray film, sandwiched to the probe bound membrane together as a sandwich, and then developing that X-ray film will give bands at the 8th step. If you observe the banding patterns on X-ray film looks like that, A and B. These are called as autoradiograms. Now southern blotting. What is this southern blotting? It's nothing but immobilization through the capillary action onto the nylon membrane or nitrocellulose membrane. So paper towels are placed at the top so that it encourages capillary action, and in this span of capillary action, the nylon the fragments of the DNA get attached to the nylon membrane. So these are cured later. The nylon membranes are cured by labeling, and then they are subjected to X-ray film to filter and observe the autoradiogram. Then applications or uses of these Rflps. These are helpful in identification and isolation of any gene known to be linked with an Rflp locus. These are codominant markers enabling heterozygotes to be distinguished from the homozygotes, as I have told. The method is simple, has no sequence specific information is required. We have not talked anything about sequence here. They used to measure genetic diversity between different populations or related species, and also important in studies on evolution. Then Rflps are also used to supplement the regular plant breeding protocols through indirect selection, used as a marker in chromosomal mapping, a diagnostic tool for diseases, are some applications of this Rflp. Then what are the limitations? It requires a relatively large amount of pure, highly pure DNA. It's a good supply of probes that are reliantly detecting the variation. The laborious and expensive task to identify a suitable marker, restriction enzyme combinations for genomic or cDNA libraries. It's time consuming as they are not amenable for automation and requires expertise in autoradiography because of using radioactive labeled probes, and also working with radioisotopes is a little harmful. Then coming to the second marker, that is a random amplified polymorphic DNA, RAPD. It's a PCR based molecule or marker technique. Here, a single short oligonucleotide primers are arbitrarily selected to amplify a set of DNA segments distributed randomly throughout the genome. RAPD amplification is performed in conditions resembling those of the PCR using genomic DNA from the species of the interest and a single short oligonucleotide primer, usually of 10 base sequence. The DNA amplification product is generated from a region that is flanked by a pot of 0 base pair priming site in the appropriate orientation. Then the genomic DNA from different individuals often produce different amplification patterns, which are referred as RAPDs. A particular fragment generated for one individual but not for the other represents DNA polymorphism, and it can be used as a genetic marker. A DNA sequence different between individuals in a primer binding site may result in failure of the primer to bind and hence the absence of the particular band among the amplification product. So, this is how presence and absence of the bands occur. The reaction products are conveniently analyzed on the agar cells, which are strained with ethidium bromide and seen under UV light. So this is the stepwise procedure. So we have two samples, sample A and sample B. So we have amplified DNA polymerase, short universal primers we have used, and we have allowed for PCR to amplify, and the amplification products are subjected to gel electrophoresis, and we have got this kind of banding pattern here. So the amplified products, if you observe the lane A for species representing sample A, we have 3 bands here, whereas in the lane B, so representing sample B, we have only 2 bands. There is absence of the second band which was observed in the species A. In this way we can differentiate these two things. Two samples of DNA can be separated. Then in inheritance studies, the amplification products are transmitted as dominant markers. Thus the presence of RAPD band corresponding to RAPD dominant allele against absence of the band that corresponds to a recessive allele. So thus heterozygous and homozygous dominant individuals cannot be separated with the RAPD markers. This is going to be a biggest drawback with RAPD markers. In F2 segregating populations, which may be scored as follows. If the band present AA, that's called as a homozygote dominant, capital A small a, which is a heterozygote, the band is absent for small a, which is a recessive homozygote. Therefore, in the F2, these two classes would be expected to segregate in 3:1 ratio, and this causes a lot of information relative to Rflp markers, which show codominance. RAPD shows dominance action. So what is the difference you observe with a codominant and a dominant marker is shown in this picture. If you observe picture number 1, the first picture, you have parent which is represented in the form of a band here, P1, and which is a dominant homozygote, and recessive homozygote parent 2 is also represented by another band, and the F1, which is a heterozygote as expected. So it is having 2 bands which corresponds to 2 parents. This is possible with a codominant marker. So what happens with the dominant marker you observe in the second picture. So parent 1, we got capital B, we got a marker, we got an amplified product here, and the homozygous recessive, it confers absence of the band as we have discussed with the RAPD case. So, and finally F1, which is also we are getting a heterozygote, but the absence of the band case is followed again here also. There is only single band, the F. So this is a disadvantage with the dominant and dominant marker. Then what are the applications of these RAPD markers? The construction of genetic maps, mapping of the traits, analysis of the genetic structure of the populations, fingerprinting of the individuals, targeting markers to specific region of the genome, identification of the somatic hybrids, a useful system for evaluation and characterization of the genetic resources. And advantages of this RAPD: it's a simple, quick to perform technique, requires relatively very small amount of DNA, and involves no radioactivity. What is the limitation? It is unable to distinguish between a homozygous and a heterozygous kind of thing. So, short differences if you observe. The principle of Rflp is a restriction digestion, whereas it is RAPD, it is DNA amplification. Inheritance, it's codominant. RAPD is dominant. Detection technique followed southern blotting here and DNA staying with the RAPD. DNA quality required is relatively pure in case of Rflp. Crude DNA is also works with RAPD. DNA amount, it's high at Rflp, 2 to 10 micrograms, milligrams. Then coming to RAPDs, it's somewhere around 15 to 25. Then primer required. No primers are required in the Rflp, whereas yes, requires random primers with RAPD. Radioactive isotopes, yes, for probes for labeling. In Rflp, it's not required in RAPD. The type of probes, SPS specific probes are used in Rflp. RAPD, it's random based. So use of primers. So probes are used here with Rflp. Sequence based primers are used in RAPD. Time sequence, 5 times more time than Rflp takes. So RAPD takes less time. Then coming to our next topic, that's microsatellites. This is the third type of marker. So what is a microsatellite? It's a simple sequence repeats of 1 to 6 base pair length. Then classification of these microsatellites as a simple microsatellites or composite microsatellites. Simple microsatellites contain only one kind of repeat sequences, as I have shown here. The first one, GT, multiplied a number of times, means GT, and so on. That's it. And the second type is AC. So simple microsatellites only can one kind of repeat sequences are seen here. Then coming to composite microsatellite contains more than one type of repeats. Then molecular basis of microsatellite polymorphism. So slippage of the DNA polymerase is believed to be the major cause of the microsatellite variation. So slippage of DNA polymerase in the sense while it is going for replication, DNA polymerase, because of slipping, that resulting in the repetition of these bases. So the mutation rate can be as high as 0.1 to 0.2% per generation. Then this is abundant and even in distribution. So what is abundant? Abundance varies with the species, but all species studied to date have microsatellites. In well-studied mammal species, one microsatellite exists in every 30 to 40 kb DNA. Then even distribution on all the chromosomes, on all the segments of the chromosomes, with the genes, often in the introns and in exons as well. So exons are nothing but coding sequences. Then small locus size, adapt them for PCR. So the locus size is very small, that's why they are adapting for PCR. Then microsatellites are codominant markers. So that's an advantage. It can differentiate a heterozygote and a homozygote. Then Mendelian inheritance of microsatellites. They are inherited as codominant markers. Then advantages of microsatellite markers. These are abundant in nature. These are evenly distributed, highly polymorphic, small loci, and these are codominant. Then development of these microsatellite markers, how they are developed. So what we need, SSR containing clones, sequences of the flanking regions of the SSR, these are needed. So what is the methodology followed? The isolation of the genomic DNA, digestion with one or more 4-base specific restriction endonucleases, size fractionation by gel electrophoresis and selection, enrichment of the microsatellite sequences, cloning and transformation, screening for specific microsatellites by cloning, colony hybridization, then isolation of the positive clones and sequencing the inserts, design a suitable primer, flanking microsatellite repeat, and test for polymorphism, and also find possibility of multiplexing. The microsatellite enriched small insert libraries are made. How they are made? The genomic DNA is digested with several 4-base pair blunt enders. And then the gel fractionation of a fraction of 300 to 600 base pair is now ligated to a phage-mate vector. So we got a small inset of DNA libraries. Then these microsatellite enriched libraries are then subjected to using a CJ236 strain of E. coli, converted into single-stranded phage-mates using helper phage M13 phage. So using a M13 phage, these are converted into a single-stranded phage-mates. So and finally, one single-stranded phage-mate which is having microsatellite is our area of interest. If you observe this, the microsatellite enriched DNA libraries, so ligation to a phage-mate vector. So we got a microsatellite into this one of these inserts, and then that what we are going to do is we are subjecting for conversion into a single-stranded phage with using the helper M13 phage. And all those things which are not carrying this microsatellite won't be converted to a double-stranded, which will be degraded in the wild-type host. So this E. coli, what is the specificity of this E. coli CJ236 is that it is a DUT-UNG among other things. So what it means, DUT, deoxyuridine triphosphatase, which will degrade the DUTPs. Because U doesn't belong to DNA, you know better, uracil is not present in DNA. Because this mutation makes non-functional DUTP will accumulate and will be incorporated into the DNA. And UNG, uracil N-glycosylase, this enzyme cuts out the U's from found in the DNA, promoting the base excision repair of the site. However, this mutation makes non-functional UNG, so U's stay in the DNA. So this is the specificity of that. Then now we got what it is, a single-stranded phage-mate we got, which is carrying the microsatellite. It is then converted into double-stranded using cancer agent 15, and then the double-stranded microsatellite enriched thing is transformed into wild type of E. coli. So this is how they are made. Then microsatellite enriched libraries, you observe this 2 base pair, 3 base pair, 4 base pair, 5 base pair, and so on. So we can enrich them in the form of libraries. Then characterization of these microsatellites. Isolate the plasmid DNA, sequence the clones, identify the clones with enough sequences for primer designing. Then, so once again, what are the microsatellite characteristics are? These are highly polymorphic, these are locus specific, these are highly reproducible, PCR based. Automation is possible with these microsatellites. Then these are the regions of simple sequence tandem repeats that lend themselves to length polymorphism, most likely because of slippage during DNA replication. The net result is that it is easy to find polymorphic microsatellites in which different alleles have different number of repeat units, so that total length of a given microsatellite locus may vary. If you observe this, so different repeat sequences are observed, and when they are subjected for gel electrophoresis, you see the variation in the bands. For a given locus containing a microsatellite, PCR primers specific for that locus are designed from unique sequences flanking the repeats. Thus, microsatellite alleles are usually based on length polymorphism. The polymorphism at the priming sites would result in loss of the bands rather than change in the length. So, length polymorphism with microsatellites can also be determined using southern hybridization using the probes specific for unique flanking regions. Then these are tandem repeats of short units of DNA that occurs with high frequency throughout the genomes of many organisms. A tandem repeat is defined as a repeated end-to-end duplication of a core DNA sequence. The microsatellite loci have a high degree of variability that is due to the high rate of mutations that alter the microsatellite length. So these stretches of DNA that consist of tandem repeats of simple sequence of nucleotides, for example AAT, repeated 15 times in succession. So a satellite is a highly repetitive DNA sequence which with each repeated sequence ranging from 1,000 to several thousands of base pairs. So the entire satellite can be up to 100 million base pair long and tend to occur in the regions of heterochromatin, tightly wound regions of the DNA that are usually not actively transcribed, found near the centromeres and telomeres, among the and also sometimes among the other places. So chromosome, centromere, and telomere regions. Then satellites are abundant on the Y chromosome, which makes the handy tool for studying paternal genetic transmission in mammals. A mini satellite is an array of tandem repeats with each repeat ranging from 9 to 100 base pairs, but most commonly around 15 base pairs. A microsatellite on the other side is an array of very short repeats, 2 to 6 base pair each, with the entire array ranging from 10,000 to 1 lakh base pairs in length, and they have so far been found in the euchromatic regions of the vertebrates, insects, and plant chromosomes. The number of repeats varies among the individuals in the population, making microsatellites particularly useful in population genetics. The entire array is usually 500 to 30,000 base pair long, and these are most commonly found in the euchromatic region, as I've told. Using the polymerase chain reaction, these repeats can be easily amplified, and the number of repeat units that an individual has at a given locus can easily be resolved using polyacrylamide gel, PAGE, that is called as PAGE. So, in a single capillary, when markers of multiple base sizes can be electrophorested together, up to 4 fluorescent dyes can be used in the same PCR reaction, enabling several microsatellites to be studied in a single run. That's called as multiplexing. So, then levels of variability detected in 6 major crops with microsatellites, if you observe. Tomato, it's having number of alleles 1 to 5. Soybean 11 to 26, rice 5 to 11, barley 3 to 37, maize 2 to 11. So this is how different variability is detected with the microsatellites in 6 major crops. Then what are the advantages of microsatellites? Highly reproducible between the labs and large number of polymorphisms per primer set. Often multiple alleles in a population, which can be highly informative. Codominant, relative abundance, extensive genome coverage, ease of detection with a PCR, small amount of DNA required, can be automated. The disadvantages: extremely difficult to find good microsatellite markers in any species. Creation of a small insert genomic library in species is required.